So we're going to talk about solving medical mysteries and what it means to be undiagnosed. This is myself and Dr. Bernstein, who will be coming up shortly. So I want you to think for a moment when the last time it was you had a fever, and think about how you felt that first day or first time when you noticed, I don't feel right. I feel warm, like temperature is not right. It's cool in this room today, but somehow I feel hot. And so to many, that's that first moment of undiagnosedness. But typically, you've met one of these guys. So maybe it's the flu virus, or maybe it's strep throat, or something that we sort of know the expected course of, where you're going to feel more fevers and maybe some chills and some cough, and maybe you go and get antibiotics, and a week or two later, you're back to where you were, and that initial moment of I feel sick has passed. If you're unlucky, maybe it's this, and this is a biopsy from a lymph node, and this is Hodgkin's lymphoma. So this is a rare disease, but luckily we have Dr. Google, and so we can go to Dr. Google, and we can type in Dr. Google what our doctor has told us. And we can even type the word treatment. And within 9 seconds, you've gone from having a diagnosis to having people advertising towards you, have a treatment plan, having other people that you can connect with. So this is also not, you started undiagnosed, you have a diagnosis, and you have a treatment. I want to tell you a story, and we're going to tell you stories of a few patients tonight, of a patient who wasn't so lucky. But his story starts the same way. So he's a 60-year-old guy, and he noticed a fever. First day, maybe it's the flu, maybe it's a cold, but it kept coming. And what came at first was fevers pretty consistently over a period of weeks and then months. And eventually he talked to his local doctor and said, I've been having these fevers, something's not right, this is different from before. They do a bunch of testing, don't find anything significant. And then he gets to come to august locations like Stanford, UCSF, the Mayo Clinic. Eventually, he's gone through a whole series of tests to try to figure out why he's having these recurrent fevers, why he's having this recurrent rash. He's undergone multiple biopsies, procedures, imaging tests, laboratory tests. He's given entire body's worth of blood over the course of a few years at this point. And he's been tried on a bunch of things that you might give to people with fevers. So many times you go to your primary care doctor for a cold or a sore throat. We're not exactly sure if it's a bacterial infection or not, but we're going to give you antibiotics at some point. If you have fevers for a long time, maybe that's tuberculosis, so maybe you get some therapy for that. Or maybe it's an inflammatory condition, so we're going to give you some corticosteroids or immunotherapies. But eventually the patient's like this. And frankly, the physicians are like this too, because we've tried things and we're not getting the solution. So fast forward a few years, 10 to be exact, and now we have the same patient, same presentations. He's now a 70-year-old male with intermittent fevers, this rash that comes and goes, myalgias, and now generalized weakness that's been ongoing for 10 years. This is what undiagnosed is, and this is the type of patients that the Center for Undiagnosed Diseases and the Undiagnosed Diseases Network across the country is really trying to help solve and try to end this sort of diagnostic odyssey. So when we see a patient, currently this is all through NIH-sponsored network, the Undiagnosed Diseases Network, and oftentimes the first visualization of that patient is this stack of records. And so these are all the tests and all the consultations and all the laboratory results from a patient that was seen at our site. As we mentioned, as Nora mentioned, that there's 12 sites across the country that are part of this network. There's many additional academic centers and centers across the world who have similar programs in place, and there's an international network of these networks that's been sharing information and optimizing the care of these patients over the past decade or so. So currently the Undiagnosed Diseases Network is operational in its 5th year essentially, and we've received over 3,200 applications from across the country and actually across the world. And we've evaluated over 1,000 and found a diagnosis in about 28% of the patients that have been seen to date. Referrals to the smaller version of the network that was through last year came from all over the country. The green dots are those who've been referred and seen at Stanford. And we're very excited, and if you would like to read more about this network, there's an article in the New England Journal led by a former graduate student of ours, Kim Splinter, and you and Ashley, one of our co-PIs, is a senior author on this paper. So from there, Dr. Bernstein is going to take over and talk to you a bit, and then I'll tell you about what happened to our patient who's 70.
Thank you, Matt. Thank you for the introduction, Nora. Happy to be here. So carrying on where Matt left off, you've gotten a sense of what undiagnosed can mean. It can mean many things, and that we're fortunate to be participating in this national network trying to help patients with these chronic undiagnosed symptoms. And a sense of so what we've accomplished up until about a year ago is what I'm going to go to next, and then we'll be actually talking about some more patient stories to give you sort of an inside look of how it is in some of these cases we are able to make a diagnosis in a quarter to a third of cases. So looking at our first 20 months, we hadn't gotten quite as far as we have as of this morning, but in the first 1,500 applicants, 600 evaluated. Who are the people who are most commonly coming to us? The most common group of patients have neurologic symptoms, be that weakness or numbness or disorders of development, things like seizures in children. The first group of people, there was 35% were diagnosed. That's at 28% right now. In that group, so some people get diagnosed and they have something that's very rare. I'll talk about what that means in a minute. And 31 participants were diagnosed with things that hadn't been described before. So if you think about the scenario that Dr. Matt was just talking about, Dr. Wheeler, so you get your diagnosis and you go to Google and you get a blank page. So it continues to be an experience filled with novelty. We don't do this alone, obviously. You saw all of the sites across the country, the labs, and the other clinical sites. Here at Stanford, we're also not doing this alone. At the end, we'll acknowledge some people more specifically. But we're a team of physicians and coordinators, genetic counselors, people working on informatics. I'm going to talk about a little bit of the computational science that is behind some of what we do. And we're really fortunate to be part of a network and to be at a medical center like Stanford Health here because it really takes a team to do these sorts of things. So when someone comes to see us, either after a few months or many years of symptoms, what does that look like? So Matt showed you the first glimpse that we get. We get an application. Often we get it in electronic form. Sometimes it is a large quantity of paper records. And all of the applications are reviewed. They're reviewed by more than one person. They're discussed at a team. If there's a decision to accept, it's discussed on a conference call where all of the sites from across the country participate. So everyone has a sense in real time about what's going on. And often people will say, oh, I've seen a patient like that, or here's some ideas. And then patients will come. They may come and see us for a series of outpatient visits, especially if they're local. They may come. Some patients may have such complicated situations they may be admitted to the hospital for a period of time. That's a little less easy for the patients, so we try to use that sparingly. So one of the wonderful things of the study, in addition to the people and the technology, are the freedom really to pursue state-of-the-art diagnostics and really things that haven't reached the clinic yet. So we'll talk a little about types of omics, like genomics and metabolomics profiling that the patients may have. We get samples of usually tissue from patients who are willing, be it a skin biopsy or blood or urine, things like that, so that after they leave, if we get a clue, we don't always have to call them back, or we can do additional studies in their absence. Sometimes downstream, we may create, with partners in the network, an example of the patient's disease in a model organism, in a fruit fly or a fish called a zebrafish, and to try and mirror. Because ultimately, I'll talk about in a moment, we're really trying to match. We're trying to match what we see in one patient with another patient, or sometimes we can't find another patient in a fly. So this slide I included because I thought to try and give a sense of what we're trying to do, this is helpful. And Matt showed, and if you were paying close attention, there was a pile of records, and then there was a DNA helix over to the side. And a lot of what we're trying to do are to find alignment between all of a patient's symptoms and the relatively rare variation they may have in their genome that can potentially explain that. And in jargon, this is called the genotype, or the genetic variation, and the phenotype is everything else about the patient. And the diagnosis, or being able to match that set of information to another patient, comes not from only the genotype and not from only the phenotype, but it's relatively unique combinations of the two. So we're looking for what are the most characteristic or specific features of our patients, and which genetic changes might they be associated with. And then we go out into the world, be it the medical literature or databases we have or people we see at meetings, and we're asking, it's a sort of a very souped up game of go fish in a way, you might say, are there any other patients like this one? I think I covered this. In the scope of rare diseases, the United States and Europe define this a little bit differently, but they end up being very similar. So we in the United States say that if less than 200,000 Americans have a disease, it's rare, so that's about 1 in 1625. In Europe, they just say it's less than 1 in 2,000. So these are relatively rare things. Some of these are at this level. Some of them are 1 in a million or several million. Some of them we only know a few people on Earth. Presumably there's other people that we haven't found, but some of these are very, very rare. However, there's thousands of these rare diseases. So the paradox is that although they're individually very rare, there's so many of them that tens of millions of people have them, and millions of children are born with them each year. So there's really lots of work to do in this area. About 80% people estimate of the known genetic diseases do have a genetic basis. There may be some bias in terms of how we're able to discover them. There's part of this is, and this is the last decade plus, about 200 new genetic diseases are defined each year. So patients that we saw recently, if we look back at all the data we collected about them in a couple of years, even if we couldn't figure it out then, there's a chance that the explanation is in there, and it was just a matter of finding the patients that matched to make that discovery. So it's a very rapidly changing area. Today is a great day to be giving this talk, one, because you're here, and two, it's actually Rare Diseases Day. So I don't know if any of you saw in the news, but organizations all around the world focused on rare diseases were on social media and events at the NIH Day, and really trying to give the message that we're discussing tonight, that these are individually rare conditions but collectively very common, and there can be approaches that are really helpful for patients who may have a rare disease. For those who are interested in sort of the nuts and bolts of things, you're welcome. Of course, you're going to have opportunity to ask questions later. When we're looking at a patient's genetic variation, we're looking at things of all different sizes. So someday, I'm hopeful there's going to be one genetic test that we can use to look at everything from the very smallest change in a patient's DNA to the very largest, whether someone's missing a chromosome or a base pair or a piece of their genome is turned around and put in backwards. We're not quite there yet, so we use a combination of methods. But it's to give you the sense that when someone says they may have had their genome tested, that can mean different things. Many of us are familiar with companies that offer genetic testing to individuals over the internet to look at their ancestry or health risks and things like that. And those use one particular kind of test that looks at a subset of genetic variation. But other tests, like looking at someone's whole genome through a sequencing study, may look at different things, and they can be complementary. And just because someone's genome has been looked at one way doesn't mean it's been looked at exhaustively. One of the oldest ways to look at someone's genome is to look at the nucleus of a cell under the microscope. You can get one step closer by breaking open the nucleus and staining the chromosomes, and a whole slew of genetic diseases were discovered decades ago when we were first able to do that. Another generation of diseases was discovered in the last several decades when people were able to look sort of order of magnitude closer using a technology called DNA microarray, which allowed us to see extra missing portions of chromosomes that are too small to see under the microscope. They're submicroscopic. And really that torrent of discoveries that's been happening in the last few years in genetic diseases is because of what's called massively parallel or next generation DNA sequencing. And sometimes this is called whole exome sequencing, looking at the portion of the genome that's actually used and expressed. And then more broadly, it can be the whole genome sequence. So instead of trying to pull out different areas and sequence them, you can just try and sequence everything all together. So that's, by way of sort of a primer. So we're all getting familiar with how we're thinking about our patients. We're collecting all of the information about their health and their history and what they look like, how tall they are, those things. And we're collecting as much information as we can about their genome. And we're trying to put those together to find a unique descriptor of that patient that would match someone else. So here's another story. So this is a story that we met a boy. He was 9 years old, and he had been in and out of the hospital for years, having fevers and problems with his joints. So he would have recurrent fevers and swelling of his joints. It was thought it could be an autoimmune condition. His joints were being attacked. However, at some point it became clear that sometimes his joints were actually infected by a number of different types of bacteria, and was really affecting how much he could walk. He'd had so many surgeries. He was largely in a wheelchair, unfortunately. And this, it was actually one of the physicians taking care of him in the hospital took a step back and said, well, gosh, he's been in the hospital so many times and we don't quite understand what's going on. He might be a good patient to refer to the Undiagnosed Diseases Center. And sort of following the model I've laid out about gathering the genetic information and the clinical information, prior to coming to our center, he'd had some genetic testing. He'd actually had an attempt to look at all of his genes known to be associated with disease, this whole exome sequencing done. He'd had a couple of specific genes tested before that, sort of trying to take a best guess about which gene might be responsible. Nothing had been found. And he'd been seen by a range of doctors looking at his immune system and trying to figure out why he was having so many infections. He'd seen orthopedists and had a variety of laboratory studies looking at his immune system's function. So coming to the Undiagnosed Diseases Network, one of the first things we do when a patient's previously had genetic testing is we look at that data again, right? Because we talked about this really brisk pace of discovery, and sometimes just looking back, especially very carefully, can be helpful. So that was not initially revealing. We went on to look at his entire genome through whole genome sequencing, also not initially revealing. We also went and met him up in the hospital, and one of the things that struck us, in addition to having all these infections, is that he was shorter than most children for his age. He had a different shape of the bones of his face, and when he was born as a young infant, he had some changes in his fingers on his hands. So his fingers were born and they were a little bit bent, which is somewhat unusual. Because of the shape of his face, he had some issues with his airway and things that was concern for sleep apnea. He was evaluated in the Craniofacial Center here at Stanford Children's, and he had a CT scan done. And what was different about his skull compared to other children his age is that people may have had a child or a baby nephew and noticed that infants usually have the soft spots on the top of their head, and these are connected to these things called sutures, which are where the bones in the head are separated, and those usually don't fuse until you're much older than this patient was. So his skull is very smooth. There aren't any of these normal boundaries or sutures, so everything is connected. And so that was kind of unusual as well. So seeing all of this, we had a really expanded sense of what his phenotype was, and then our team went back and very carefully went back and looked at his genotype again, sort of a third pass, actually. And this time, two changes in a gene called IL-6 ST were noted. And one can come from his mother and one came from his father. So it's consistent with a family of conditions called recessive, where you have a change from each side of the family. And each of these changes were exceptionally rare, and they'd never certainly been seen in combination. And importantly, in the period from when he first had his genome sequenced to when this was noted by our team, two other patients in the world had appeared in the literature. They'd been seen by doctors who had made very similar observations. And this is what they looked like together. So there were patients in Europe, actually, and they had these very characteristic things that were similar. So they had the fusion of the bones in the skull, which is less than a 1 in many thousands of children would have that. They also had this contracture or bending of the fingers. They'd had the fevers. They'd had infections, mostly of other parts of the body than the joints like he had, but some had erosions of the joints. And they were all small. And then when you looked at their laboratory tests, they had similar differences in their immune system. So there's a whole bunch of highly unusual things that are all shared in constellation amongst these patients, strongly suggesting that these are amongst the first people in the world to have this particular condition, which doesn't quite have a name yet. It's an IL-6 related immune deficiency with elevated IgE, one of the types of antibodies in our body. So because our patients' genetic changes had not specifically been seen before, we did work with a laboratory that had seen some of these initial patients, and they tested the patient's immune system in the laboratory and showed that it wasn't functioning in a specific way. So this gene is an instruction for a protein, and it's part of a system for how cells and the immune system elsewhere communicate with each other. And it has two parts, interestingly. So there's, if you will, there's a part that's on the right, or my right over here, and then on the left. And one seems to be very important for the bony differences we see in our patient. And in fact, there's some patients that have only a problem with that part. And the other part seems to be the other side of the pathway, if you will, is because it affects the immune system. So there's also patients who have only differences with the immune system with related genes. But these three patients had problems with both sides because of where this protein functions in the signaling. So it was very rewarding for us as a team. And this is an example of a patient where it's really changed the whole view of how we think about his condition. So he wasn't someone that was inexplicably having many infections, and he wasn't someone whose immune system was activating for no reason. There actually were differences in immune system and infections, and we've got him on his way to seeing some specialists who hopefully will have some new ideas for how to take care of him. So this is another story of a child about the same age who came to see us with a completely different set of symptoms who were able to help in a different way. So the genetics end up being pretty different here. So he was born and recognized to have some differences at birth as well. He had a lot of differences with the veins in his body. So yeah, this is a great screen we have here today. So for example, a part of one foot is sort of much redder than the other. He's got one hand that's much redder. It's because there's a lot of really prominent capillaries there. He's also got some large veins, very prominent. This is on his back coming through. That turns out that's because he's actually missing one of the main parts of the vena cava, the large vein that helps the blood return from the lower extremities. So the blood is going around the missing part. And he's got these really large veins on his back. And then in addition to having the abnormal veins and capillaries, he's got some areas where there's extra pigmentation in his skin. So there's extra melanocytes you can see on the white of his eye, actually just 1/2 of the roof of his mouth, which is a very distinctive thing. In the last case, the patient had this fusion of the bones in his skull, which was quite unusual. This patient has this unusual distribution of unusual skin findings. Before he came to us, he had not had any genetic studies because of the conditions that were being considered for him. None of them had a known genetic cause at the time. He had had this picture taken, an MR angiogram of his venous system, and it shows that he's, you can see the veins coming up out of one side of his leg, but coming out of the other, there's no normal flow here. So it's coming around, and that's where you see this large vein superficially on his back. And he'd been seen at many centers around Northern California, weren't able to come to a diagnosis. So we saw him and examined him, and in thinking about him and the combination of symptoms that he had, he actually did match a group of patients that has been reported in the medical literature for decades called phacomatosis pigmentovascularis, very long name. And it basically refers to patients that have these differences in the capillaries and in the pigmentation. But the cause wasn't known. And we thought it was worth going to see if we could figure that out. Probably we were a little bit foolish to try something because it's not like other people hadn't tried. So we had his exome sequence from his blood, and it didn't give us any clues. But because of the patchy nature of his findings and the asymmetry, we considered the possibility that he could have a difference in his DNA that was only in part of his body. So if we all start as a single cell after we're conceived and it starts dividing in half, quarter, and eighths, if you have a change in your body in the DNA that occurs after conception, can affect only parts of the body. It's a phenomenon called mosaicism. It refers to art made from tiles that's mosaic in different places. So he had DNA taken from skin in the area where it looked different, and that was compared to the DNA from his blood. And then there was a change. It was one that was very hard to see. It was present only in sort of 5% of the sample, and it was in a gene called GNAQ. There's not a quiz on the names of the genes at the end of this for sure. And again, what was really helpful is sort of that on its own is interesting and suspicious, but it's really having another patient. And again, lots of activity going around in this area. Another group had found a handful of patients that had very similar symptoms. Each patient with this disorder is a little bit different because it's random which parts of the body are affected. But they had very similar differences in the skin pigmentation, the deep veins. Many had differences in the central nervous system. And they all had actually one of a very few changes in this gene because it doesn't break how the gene functions, it actually turns it on in an unusual way. So again, it's the combination of the very specific, unusual clinical findings and genetic findings that together form a diagnosis. This one is different than the other one because it was this mosaic condition. The third case I wanted to talk about is another child. I'm the pediatrician here tonight, so it's all children. Matt will finish with the adults. But this is a lovely, really charming. She was 7 when we first met her several years ago. I think she's 10 or 11 now. You may, if any of you have seen our work, her family has been very generous and participated in some interviews we've done with local media in the Bay Area. In any event, she had a very rough start to her life. Shortly after she was born, she was in a coma. She had a huge amount of extra acid and ammonia in her blood. It's really the ammonia that is like a depressant to the brain. And then she received intensive care therapy and recovered from that. But periodically throughout her life, whenever she got really sick and couldn't eat or had a fever, the stress would cause this derangement of her body, and her metabolism would go way out of whack. She'd get very sick. At health, she's very active, but she does fatigue easily. She's got a just lovely person. She's a little bit smaller than average for her age, and she's not nearly as strong as many kids her age. The type of extra acid in her blood is called lactic acid, so we all get some lactic acid in our blood, so our muscles get sore when we exercise a lot. And that was suspicious that there could be a problem with how her body made energy. So there's a whole family of conditions that affect the mitochondria, which are part of our cells that make the energy that we use to move. And there was a suspicion she could have something like that, but it was really hard to pin down. Because there was so much suspicion for this, she had her mitochondrial genome sequenced. She had her exome sequenced. She'd been seen by many, many doctors. She'd had biopsies. She'd had tests of enzymes that could be deficient in her body, MRI scans. She had problems with her heart. She had an echocardiogram. But there wasn't, there were some very specific features in her phenotype, but there was nothing in her genotype that would allow us to give her a specific diagnosis. So we only had sort of half of the equation, so to speak. So the next step for her was looking again is really important. So looking again at her exome sequence, the team here looked very carefully, and they noticed that there was a change in a gene that had never been associated with human disease before. It's one of the components of the energy generation system in the mitochondria. It has a not very memorable name. At the time, it was called ATP5D. And the change she had was also sort of hard to tell if it was important. Some changes, it's very clear they break a gene, and this one was suspicious because it was so rare, but hard to tell. So a fruit fly, which I'll show you in a minute, was made that had this change, or in a way had this change. And we went to look for other patients. So we took all of her data and we put it into databases anonymously to see if we could find another patient. Matt went up to a meeting, I think in Vancouver, and actually ran into a physician from the United Kingdom that had another patient that seemed similar. We also profiled her metabolism in more detail. So Mike Snyder's lab in the Department of Genetics here at Stanford is leading a project looking at what's called Metabolomics, looking in very high detail at the metabolism of children. And when they looked at the flow of metabolites, so the chemicals in our mitochondria that help generate the energy in our cells, they saw that there was like a backup. Sort of like if a lot of traffic's trying to go through a part of the road where it's really narrow, all the cars behind the backup build up. So there's many parts of this diagram, but the one part of it is called the citric acid cycle, and many of the substances in the cycle were building up, suggesting that there was a block downstream. You sort of follow the arrows, through here, and there was a block way down here, and things were piling up behind it in our metabolism. So this was starting to get very suspicious, in that there was our patient had a rare change in a gene. She had unusual symptoms. She had buildup in metabolites that fit with a problem with that gene. There was another patient that we were exploring with scientists around the world to see if they had the same thing. And a group of scientists through the UDN, through the Model Organism Sequencing Center. These are scientists at Baylor College of Medicine, led by Mike Winkler and Hugo Bellin, made a fly. So this, you'll believe me, is a regular usual-looking fly. This fly has a very small head. So if you take this gene that was different in our patient, subtly, very subtly different in our patient, out of the fly's head, it takes a lot of sort of trickery to do that, it has a dramatic effect on the fly. So clearly this is an important gene, but we're not looking for a patient with a very small head. We're looking for a patient with an intermittent set of symptoms that affects her energy generation. It also affected the eye. So if you take this gene out of the fly's eye, it doesn't have the sort of usual appearance. It gets sort of smaller and smooth. But if you put the regular version of this gene back into the fly, it fixes all this. And if you put our patient's version of the gene back into it, it fixes it some, but not all, suggesting that there's a very subtle deficit in our patient's gene function, which would make sense, because if you think, well, if your body can't generate any energy with mitochondria, that's probably not compatible with walking around on Earth. But if you had a very subtle difference, it might just cause problems when you were stressed. So it fit together. So Matt went to Vancouver. Many of our patients create web pages about themselves. We help them do this, obviously with their permission, to try and find other patients. And there are actually data sharing networks that exist in the cloud, so to speak, where patients and doctors from around the world put what we believe are the specific aspects of their genotypes and phenotypes out for other people to find, with the idea that they'll match. And periodically we get notified by these systems that there's another patient out there that could be a match for our patient. And this is increasingly how this is happening. And it's really, that's part of in addition to the sequencing, that's why this is accelerating so fast, is it doesn't require Matt being in Vancouver, although it still happens that way sometimes. We can both be sitting at our desks in Palo Alto and be in touch with someone in Australia or Japan or Europe. So that's sort of a sense of the type of cases we've been able to take on. So a lot affecting energy generation, the development of the body, the patterning of the skin and pigmentation, the immune system. All of these things can be genetic. But we'd really like to take it to the next place. How can we do this even better? So one of the areas we've been working on is how to be as efficient as we can about this, looking at our data, which currently is a really painstaking process. People have estimated that it can take up to like several days of person time to carefully look through all the genetic variants in a patient. And increasingly this is supported by computers and it's getting more efficient. But we imagine a day where the person would only have to look at the genetic changes that the computers tell us are the most important. On the other side, we also want to be able to gather the information about the patient's phenotype as efficiently as possible, even if that's in the medical record. So we're developing systems that can extract information from the medical record automatically, efficiently process the patient's genetic information, and also pull all of the knowledge out of the medical literature and bring those things together in an efficient and effective way. So this is important just for being efficient, but this is also really necessary for us to continue to do our job, is sort of what we've come to believe. Because as I said, these conditions are individually rare but collectively common. They are affecting millions of people in aggregate. And it's becoming possible for probably most of these people actually to get their genome sequenced. So right now, it's still relatively difficult to do. Not all insurance covers it. It's looked at as sort of an unusual thing to do and expensive. And a decade, 2 decades ago, the first people's genomes were sequenced. It cost nearly $1 billion. It's come down through the hundreds of thousands, tens of thousands. Right now, it's on the order of a few $1,000s, and it's in the news that there's the hope that current technologies would take it to about $100 for just the technology alone. So with this happening, the idea is that millions and millions of people with possibly rare diseases and even healthy people are going to be in their genome sequence, and there probably aren't enough of us altogether to be manually looking through all of that data. So it becomes not just convenient, but necessary to automate this. So a few of the ways in which we've done this in collaboration with folks in computer science here at Stanford, especially the lab of Dr. Gil Bejarano, are developed tools that do this. So Amelie is a package of software developed by Dr. Bejarano, work led by a graduate student, Johannes Bergmeyer, that does this. It goes out, looks at the medical literature, tries to decide which articles are about genetics and which are about genetic diseases. It's able to access those and pull out of them all of the information about relations between symptoms and diseases. So exactly what we're trying to know about in diagnosing these patients. So right now we work with books and articles ourselves and databases where people have manually extracted all this information, but there's so much of it, there's the hope that tools like this can help us do it automatically and be even more efficient. So this is a sample of patients' data, but you input the set of genes you might be interested for a patient, their phenotypes as codes, so the same two things, the pile of papers and the piece of DNA, and then the computer will look into its compendium of all of the articles that it's extracted and try to point out to the clinician which articles and therefore which genes are most relevant to the case. And it's able to do it reasonably well. It's certainly well over half the patients, in some cases up to 60, 70, or 80%. It does really well. It gets the right gene in the top. Obviously, it, as we do, struggles with the hardest of cases, but we're trying to push this forward. So that's the medical literature part of it. Also trying to automate the extraction of the information from the patient's records. So patients come in with thousands and thousands of pages of records. It can take a long time to go through that. You might want a very efficient way to pull all the information out. The other idea is if you're going to go back and look at the patient's genotype again, you might want to go back and look at their records again. But you don't. If I follow several hundred or thousand patients, I can't be spending time every week looking at all of their records. But a computer can do that. So it can be monitoring a patient's record, looking for an important change that might be the clue that is the breakthrough to what their diagnosis is. So this is a tool called ClinFen. This was, again, Johannes Bergmeyer and now Stanford student Cole Diceroth. And they developed a system to automatically pull the information out of the patient records. And the way they do this is they take the records as text, they break the sentences down into groups of words that seem like they should be together, and then they process those and try and see which standard terms they may match the most. And they do really important things, like they don't pay attention to things that the note says the patient does not have. They try to ignore things that like the patient's uncle has. So they just want to know what the patient has. And depending on your view on technology, it's either surprising or expected that it does this very well. So it pulls things out very quickly. And some of the really brilliant work that they did is that their system functions at takes about the same amount of time to look at records from like 100 pages to 1,000 pages, whereas other systems slow down a lot. So it's very efficient. It can take on data from a large number of patients at once. And when you try and do things like rank the patients by how likely a diagnosis is based on the input just from the ClinFen tool, it does nearly or sometimes better than how a list of terms from a person would do. So there's a lot of promise that this can help us automate a lot of this work. And so we've talked about looking at the patient's data initially, reanalyzing it. Earlier I showed you the 200 plus diagnoses new conditions being defined each year. And what we estimated is that because of that, you just wait a couple years and 10 to 15% of your undiagnosed patients will become diagnosed just by looking back at their information. So we've had success with this approach on a number of patients. Every patient who has their genome sequenced here at Stanford through our clinical genetic service, we give the option to have their data periodically reanalyzed, both through a clinical lab that did the testing initially and also on a research basis to give us the best chance of, as close to that moment as possible when the information either about them or about their genome, what we know about it changes, that we can see that there's been an important change, and we'll find out and let them know that there's been a diagnosis. It's a little bit of an unusual experience as a patient, and that usually we expect we'll get information from our doctor when we go to see them. And this is sort of something that's just in the background. So to me, I was driving home one day, and it occurred to me this is a little bit like the warning lights that go off in your car, and that you can be driving along just on your regular commute. I have patients I haven't seen in 5 or 6 years, and something will turn up in one of our systems or at a lab where they were tested, and suddenly what didn't make any sense will be a diagnosis for them. So it's really wonderful that this can carry forward. So I'm going to turn it back over to Matt, who's going to wrap up, and then we'll look forward to your questions.
So we're just going to go back to thinking about our adult patient for a second, and think about the story in him in light of what could be found. And I think, as Dr. Bernstein mentioned, for each case, it's really important to think about the phenotype. And sometimes it's not only important to think about the phenotype, but to get the right person thinking about the phenotype. And so our patient comes into the Undiagnosed Disease Center at Stanford, and we had him see a few different physicians. And one of them is a really excellent colleague of ours who's an infectious disease doctor, and said, I've read this before. I've heard of this story before. And so, again, 10 years after presentation, in this case, really the right physician getting the right information. And as in many of these other cases, there is one new piece of information that sort of made this more likely, which was a fairly common, upwards of 1% of people, as they get older, will develop what's called MGUS, or monoclonal gammopathy of uncertain significance. In this case, it was not uncertain, and it was very significant because it led to this diagnosis of Schnitzler's syndrome. So I expect that all of you have read a lot about Schnitzler's syndrome, as I had at the time. But it is a constellation of things, and like many other syndromes that are described in the literature, it has a bunch of things, and you have sort of degrees of criteria and degrees of certainty of diagnosis. But he basically met almost all of the criteria and met the criteria for a definite diagnosis of this Schnitzler's syndrome. And so in this case, Dr. Google was available and did answer our house call. And we were able to see examples of other people who had the sort of evanescent rash in the same way our patient had. And even a summary of sort of the typical findings. But what to a large part was missing from this literature is sort of the why. So what is happening and why? And there were some hints, but the why of this diagnosis was not really clear from the literature at that time. And so we had the opportunity to use some of the technologies that have been developed at Stanford over the years to take a look at using this patient's unique biosamples to say what might be the why. And so one of the ways we can look at somebody's immune system is through profiling the cytokines, or circulating molecules that lead to fevers and your immune cells moving to one location or another. We can look at the makeup of the immune cells. So everybody has sort of a constantly shifting group of immune cells, and so we can measure those exactly, and we can measure those at different time points. And then using a technology, again, the lead development was done here at Stanford to basically measure the signaling within those immune cells, and we can actually measure each of the immune cells we look at and the signaling that happens within those cells in response to cytokines that might tell those cells to become more active or less active. So just one example is that this is cytokine profiling of this participant along with other participants at Stanford. You might notice that most of these patients have relatively modest changes on our scale here, and then there's these two samples at the top. And these two samples, which are where this cytokine is much more evident in his blood, are actually a day apart. So he came in, and then he had a fever, and then we got another sample, and we can see that there's increase in this cytokine in response to that. And so this information combined with some additional information in the literature actually led to a change in the therapeutic approach to this patient. And so he was switched from one type of immune system modulator to another type of immune system modulator that was felt to be more closer to the source of the problem. And in this very large study in Schnitzer syndrome, 3 out of 3 patients had a sustained response with this therapy. And as did our patient, had a reduction in the frequency and severity of his fevers and rashes. So, so we've given you sort of an overview of what it means to be undiagnosed, what the Undiagnosed Diseases Network is, some thoughts about rare diseases, some thoughts about why these rare diseases are so difficult to diagnose, and some of the technologies that we and others are using to both accelerate the discovery and conversion of patients from the undiagnosed state to the diagnosed state, and also opportunities to use these tools to allow the pursuit of therapies and allow for communities of these rare disease patients to come together. So we would like to acknowledge the major funders of the Center for Undiagnosed Disease, which is the Common Fund of the National Institutes of Health, and the School of Medicine, the Departments of Medicine, Pediatrics, Genetics and Pathology, and Neurology, and laboratories of our collaborators in the translational and basic science side, Gil Bajarano, Steve Montgomery, and Mike Snyder, and the 4 co-PIs of the center here. And we certainly couldn't do any of this work without our extensive team, many of whose names you've seen on our slides, and the entire Undiagnosed Diseases Network. So we'll take questions. I'll invite Dr. Bernstein back, and if there's any things that we don't cover, there's information behind each of these links that might be able to help. So. In the blue?
Yes, my husband was diagnosed with ITP 10 months ago. He's been through, as you say, there's a stack like this of scans and biopsies and they still can't get him up above 40 max, his platelets. And, you know, he's had IVIG, he's had Nplate. Would this be something we could bring to you?
So there's one of the, yes. So the question was a patient with ITP, idiopathic thrombocytopenia with purpura, something like that. Would that patient who's having difficulty with therapy treating their symptoms, is that something that's appropriate for the Undiagnosed Disease Network? One of the challenges is there are lots of rare-ish diseases where there are both people doing targeted research in that area, or the sort of the standard pathogenesis of that disease is under investigation. And so oftentimes we, if people with those sorts of diseases are applying to us, then we try to find the research group that's best equipped to help with that. So we tend not to be able to help people who have a diagnosis and a therapy, and that therapy isn't working. But it's always something where there may be somebody who, either at Stanford or elsewhere, can help. Another group of patients where our resources aren't probably the best resources to pursue is in the area of sort of cancer of undifferentiated type. There are some people at Stanford Cancer Center and at the National Cancer Institute who sort of specialize in those types of cancers, and so we do try to facilitate people getting access to those resources in their research setting. That's a great question.
How does insurance coverage play into all of these tests and other procedures?
So right, yeah, so the question was, how does insurance coverage play into all of these tests? So right now, the Undiagnosed Diseases Network is a clinical research project. And so some of the tests we show and some of the tests we do are not clinically available. Others are clinically available but expensive. And there are certain things that the Undiagnosed Diseases Network and the funding from the NIH support and fund. There's other things that are part of the standard of care that patients who come to us might not have had, and so we facilitate those tests being done, and insurers will generally cover those things. The actual process of being seen at an undiagnosed diseases center is not yet something that insurers have sort of committed to funding, and that's one of the sort of challenges for the network going forward, is to show that the process of the network works and accelerates diagnosis and or leads to better outcomes in patients.
How often do you actually come up with a therapy, or the therapy results from this work? It's kind of nice to know what you have, but if you're a mitigation or a cure, it's...
Yeah, so the question is, of patients who are diagnosed through this program or similar programs under a different organization, in our series, it depends how you define therapy. So people will define sort of hierarchically any kind of intervention, but for defining a sort of therapy, like something specific that's supposed to make a difference, it's about 1/5 in that first 2 years experience of the NIH Undiagnosed Diseases Network. So it's definitely a minority. I think that we wish it was everyone, but certainly there are many diseases for which that's the next step. So first, recognizing that there is a diagnosis is often a prerequisite for even thinking about the possibility of what a therapy could be. If you're the first person to have a disease, it's unlikely there'd be an existing therapy. There are some unusual cases where you're one of the first people and it just happens to be similar to another disease for which there is something you could essentially try on a research basis. The other... It's there's sort of two, if you think of like pillars of rare diseases research and therapy. One is the diagnostic phase and the other is the therapeutic phase. And things are changing very fast in both areas. Arguably the rate of change in therapies is even faster, even though it's starting at a lower point than the diagnosis. But there are people at Stanford working on therapeutics for rare diseases. There's a lot of rare diseases. And there are many companies. There's a whole industry referred to as orphan diseases. There's federal legislation that has provided incentives for this over the years. So there's a lot of hope for people that are diagnosed with a very rare disease, even if there's not a therapy now, and certainly for the people who might be diagnosed for years from now. So we wish it was everyone. The answer is it's a minority, but I think it's increasing. Yep.
When you treat a patient, you often have to treat the whole family. And I'm asking this in a generic sense because I'm not the least bit worried about it. But when I was in high school, my dad had a very rare disease in his eye, and it just swelled up, and he couldn't see, and it went on for a couple of years, and they took him up to Ann Arbor Hospital, a Veterans Hospital, and determined that he had a disease that only 6 other people had had. How do you relate that to the family around them when there's genetic possibilities of the same disease and so on and so on.
Yeah, so the question is about, do certain diseases have an impact on other family members, and what do we do about that? So that's something that's certainly arguably more true about genetic diseases and other types of diseases, though there's many diseases that are shared by family members that aren't largely genetic. Sometimes you can really take away a lot of the worry from a situation like that, because if you understand the genetics of the disease and what the disease is, you can be very precise about who might be at risk and who's not. So that's sort of looking on the bright side. But on the other hand, sometimes you do find out what one person has and realize that other people are at risk and might not have had symptoms yet. And patients approach this from all different backgrounds and all different outlooks, and it's really about trying to learn about the patient and the family, and what, as a partnership with the patient and the provider, what people agree on would be the most helpful to them. So some people want to know everything just for the sake of knowing. Other people only want to know things they can do something about, and other people really don't want to know anything. That's sort of trying to be a crystal ball, right? So trying to be sensitive to that, but helping people understand what the pros and cons of those different approaches are, and explaining things usually over time. A lot of people come at this type of question, and it's like, well, how do I do this right now so everyone understands everything? And most of these things need to be a conversation over time and work better if there's a relationship.
Suppose, for example, father and son, mother and daughter, whatever, walked in and appeared to have the same symptoms, but yet you couldn't identify either. It sounds like from everything you're saying, the first thing you'd approach would be genetics as opposed to transmittable disease, for example.
No, I think that, it may be, I'm a geneticist, so I have a bias. And our network is geared up and was started largely by people with backgrounds in genetics. So there's a heavy skew, I think, towards what we aim to take on, what we do take on, that it's likely to be genetic. But it can be, I imagine that it's very context dependent, and whoever's seeing a patient would use their best judgment about what's most likely. Yeah.
Request reanalysis of the exome test, and also those data matching skills like Amelie and Clint.
How do you request, was your question? So the question is for a patient who may have had through their doctor or perhaps a research study. I think it's easier to answer your question about through their doctor. So nowadays, some patients are able to get some of this genomic testing done through their doctor. And the question is, how do you request reanalysis? So different laboratories have different policies about this, but most of the larger laboratories offer, as part of giving the test, to reanalyze it at least once. Some will allow it to be analyzed annually for unspecified number of years.
What about Stanford? My younger daughter is in Stanford System. She's been seen in Lucille Packard Children's Hospital, and also the pediatric neurology here.
Yeah, I'd be happy to talk about questions about specific family members and patients maybe afterwards. But in general, these requests are made through the provider that helped the patient get the test. So you'd want to we can talk about it after, but one would get in touch with their doctor and say, I heard that this type of thing can be done, and maybe can you help me explore what the options are for that for my family? Yeah. That question?
Yes, with all these hundreds of undiagnosed of diseases and so forth that you're talking about, and the hit and miss and your research, I'm pretty impressed. But it's really sad about all these different undiagnosed diseases and so forth. Do you have any information or has there been any research in the area of the possibility of some relief due to meditation or religion or the placebo? I know Stanford's a couple of years ago they did some research in the area of the placebo that was in National Geographic, and Susan Richards does some research here on campus as well. And so there have been cases. I just don't know if you guys have done any research in those areas or about relief in those areas.
Yeah, so the question is, given the many different types of undiagnosed diseases, is there evidence about the effect of meditation or other types of religion or things like that may affect these diseases? I think this is one of the areas where when we're talking about going from undiagnosed to diagnosed, it's very challenging to prove causality of things that are difficult to measure. And so I think a combination of the past two, the question from the gentleman about non-genetic things is much more difficult to, in a single person, to say this environmental happening or stress or whatnot has impacted that person's disease in a sort of definite scientific way. And I think the second component is then thinking about diagnosis to the expectations after diagnosis. So we've talked a lot about the treatment challenges in patients with very rare diseases, but there's another type of challenge which I alluded to in the beginning of the talk, which is expectation challenge, and what is going to happen to me in the future. And so when we don't have that, because one of the cases we present, there's literally 2 people in the world with that, we don't know what's going to happen to that patient. We have no way of knowing for sure what's going to happen to that patient. So it's very hard unless a patient is having very frequent measurable symptoms for us to be able to say clearly this intervention has this effect on their phenotype. And so I think for our patients, who have diagnoses, there's still challenges, but there are different types of challenges than patients who remain undiagnosed. And so there's certain things that the network is doing to look at the sort of effects of the process of seeing one of our centers or getting genetic test results or other test results on their sort of well-being, but in terms of an intervention that's applicable broadly to rare diseases, I think that's very hard to come up with a rigorous trial to figure that out.
How many new patients is the center here able to see per month or per year?
That's a great question, and so it's partially related to our sort of staffing levels and our funding levels. But our goal in the past 18 months or so has been trying to see on average 1 patient per week, and so we're hopeful that with generous support from the departments, School of Medicine, and potentially individuals in the area, that we might be able to continue that. But the NIH doesn't have that plan for us currently, and so we're looking for alternative approaches. Yeah.
How often, with all the exotic chemicals that we have out and about nowadays, how often do you run into chemical exposure being the cause of a disease or seeming disease?
So of the diagnoses in the network so far, there haven't been any definite chemical exposure to diagnoses causative links to date. There are certainly in the history of medicine and rare diseases cases where that is the case. In our cohort, I think there's 2 cases where we have a suspicion that is in part in one case where basically 2 people that were cohabitating and not related had the same thing that was adult onset and with fairly aggressive presentation. But it's, again, for similar reasons to the answer to the first question, it's hard to prove, and particularly if there's an exposure that wasn't measured at the time, then it may be impossible to prove.
So it sounds like it would be more of a trace it through history as opposed to trace it through testing.
Yeah, I think the so the question is something you can trace through history or through testing, and certainly we can develop a suspicion or a hypothesis through a history, but I mean there are ways to show that a particular exposure leads to a biological phenomenon, but we have to connect a lot of dots to come up with that. And there is one of the sites has led the development of a questionnaire that patients get that looks at all their exposures. And so that is something that we collect, but as yet it hasn't led to a definitive diagnosis.
That looks like maybe the last question.
Thank you very much.
We'd like to thank everybody for the questions.